Hello, my dear children. Welcome to Byju's. This is Neha Sharma, and we are in the process of solving NEET 2019 paper biology section, as you already know. So, what are we waiting for? Let's begin the session. Let us see this question. The correct sequence of phases of cell cycle is. So we have been provided with four options here with different sequences of G1S, G2M. Okay, first thing, what is cell cycle? Cell cycle is basically a series of changes that are happening when a cell wants to divide, right? So it's all the processes that are happening before the cell division as well as the actual division part. And this whole thing collectively is called as cell cycle. All right. When you look at cell cycle, let me show you an image so that it becomes easier for you all. So when you look at the cell cycle, you know there are two major phases to it. First one called as interphase. All right. And the second one which is called as M phase, which you can see very well written here. Okay. So what is interphase? Interphase is actually the main preparatory phase of the cell cycle where the cell does all the preparation that is required for the cell to undergo division. All right. So as it might seem like that it's the resting phase, which was, earlier the misnomer of it. So it's not the resting phase. It's actually the preparatory phase and one of the most active phases of cell cycle. All right. Interphase actually occupies almost about 95% of the time of the cell cycle of whichever cell you look at. All right. Now this interphase is further divided into three major phases, which is G1, S and G2 phase. G1 stands for gap 1 phase. We also refer to it as, sometimes growth 1 phase. Okay. S stands for synthesis phase and G2 stands for gap 2 phase. So starting from here till here, this whole thing is what is referred to as the interphase. All right. And after the cell has prepared for division, after the genetic material has duplicated, whichever cell organelles had to, duplicate themselves, that has happened. All the spindle protein have formed itself so that the chromosomes can be nicely separated during the my M phase, right? So all these things are going to happen in the interphase. And after that follows the M phase, which is the actual division phase of the cell cycle. Clear, everyone? So obviously, now when you think of the sequence, what will be the division or rather the phases of the cell division or phases of cell cycle? It will be starting with G1 phase, then it will be followed with S phase, then comes the G2 phase, and then comes the M phase. And that is the reason why the right answer for this question is going to be option 1, that is G1, S, G2 and M phase. I hope you all are clear with that. Which part of the brain is responsible for thermoregulation? Okay, now what is thermoregulation? It is basically one of the most important aspects of homeostasis, especially now here where we are talking about humans. Okay, we know that the capacity of humans to survive in various drastic ranges of temperature is quite good. We survive from hot arid temperatures to hot humid temperatures to very cold regions where the temperature is in sub-zero level, right? So when you talk about thermoregulation, one of the most important parts of the brain is what is controlling it, which is what is called as hypothalamus. All right, hypothalamus is the part which basically controls or secretes out all these various different hormones which control a lot of bodily functions, which in turn generate body heat and which in turn maintains the temperature of the body, and that is what is referred to as the thermoregulation. Okay, now here the other options that are given are medulla oblongata, cerebrum and corpus callosum. So I would like to give you a very quick revision of the different parts of the brain and where exactly is hypothalamus located and what are the functions of these incorrect options. So starting with the parts of the brain first, you know that if you look at the overview, the brain is going to be distributed into forebrain, the midbrain and the hindbrain. Forebrain has further three major parts to it, that is cerebrum, which is like the biggest part of the brain, right, which is also one of the incorrect options that are given here. Then there is thalamus, And below the thalamus or beneath the thalamus is the next most important portion, which is what is called as hypothalamus. Okay, then we go on to the midbrain, which is obviously located at like the central position of the brain, and then going on to the hindbrain, which is the third part of the brain, which is further going to comprise of the pons or the pons varoli, like we say, then medulla, which is a part of the brain stem, right, and then the cerebellum. Clear, everyone? So starting with the hypothalamus first. So like I told you, it lies, just beneath the thalamus or at the base of the thalamus. And the most important point here is that it contains a number of centers which control the body temperature, the urge of for eating and drinking as well. Okay, so it's very, 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 very important for you to remember. Normally, the question in exam regarding thermoregulation and regarding this homeostasis, regarding hypothalamus, it's all quite direct. Okay, you usually get direct question like this based on thermoregulation, and it's a very important concept that you need to understand. Then the next important point, how exactly the hypothalamus works. The most important point here is that there are these specialized neurons which are called as neurosecretory cells, which secrete out specific hormones which we refer to as hypothalamic hormones, which in turn controls the functioning of a lot of internal organs, like you talk about liver, you talk about kidney, you talk about heart. All these important internal organs, which are usually with their physiological activities going on, are responsible for generation of decent amount of heat that is required for the bodily temperature maintenance. Okay, then we go on to the next option that was given, medulla oblongata, which is the incorrect option, of course. It's basically the part of the brain stem. Okay, please remember it this way. So it's like the stem-like structure which is present here, right? Now, so this medulla oblongata basically controls, or rather contains, the centers which control respiration, cardiovascular reflexes, etc., and gastric secretion. So there are various different bodies and receptors present in the medulla which controls all these important and vital functions of the body. And that is why it is said that this hind brain part of the humans should not be hampered, or it should not get affected, or it should not get damaged, because it basically controls all these vital functions, like centers for vital functions like respiration, cardiovascular reflexes, etc. Then we go on to cerebrum. Like I said, cerebrum is the biggest part of the brain. It basically is going to contain two major hemispheres, or rather is made up of two major hemispheres, the right hemisphere and the left hemisphere, also called as right cerebral hemisphere and the left cerebral hemisphere, which are separated by a deep central fissure. But then these two hemispheres are very well connected with the help of a band of nerve fibers, which is called as corpus callosum. Okay, so it's divided into two parts, right and left cerebral hemispheres, which are connected by the corpus callosum. What is corpus callosum? It's basically the C-shaped nerve fiber bundle that you can understand it as, right? So what you see here, this is what is the corpus callosum. Yeah, and as I'm telling you, because it's made up of this whole nerve fiber bundle, it is one of those part of the brain which has one of the maximum amount of white matter of the brain. Clear, everyone? So for this reason, all the other three options, that is cerebrum, corpus callosum, medulla oblongata, are all incorrect options for this question, and the right answer stays as hypothalamus. Let's have a look at this question. Which one of the following is not a method of in situ conservation of biodiversity? First thing, what is biodiversity? Biodiversity is basically the wide variety of organisms that are existing in nature and in consortia with nature, actually, I should say. All right. So this biodiversity needs to be protected, because it's very, very important for the nature to balance out biodiversity at all different levels of the ecosystem. Clear, everyone? So now, when you try to protect this biodiversity, conserve this kind of variety of organisms which are living with each other in nature, you can do it in two different ways. One is in situ and the other one is called as ex situ conservation. All right. In situ conservation, ex situ conservation. Very important, very interesting, of course, of practical importance. Okay. In situ conservation is when you try to conserve and protect the variety of organisms, that is the biodiversity in its natural habitat. Okay, so conservation and protection of biodiversity in its natural habitat is what is called as the in situ conservation. And when you try to conserve the biodiversity of, not just the rare plants, but for that matter even the regular plants or animals or certain kind of organisms outside their natural habitats, this kind of conservation is what is called as ex situ conservation. Okay, when you look at a few examples of in situ and ex situ conservation, this is what you can think of. Okay, first thing, let me start with in situ conservation. So like I told you, this is going to be done in the natural habitat. The few best examples of in situ conservation are sacred groves, the biosphere reserves, and the wildlife sanctuaries, exactly what you have been given in your question. Okay, there are sacred groves, there are biosphere reserves, there are wildlife sanctuaries, there are sometimes national parks also you can think about, right? All these examples are of in situ conservation, where you are protecting the biodiversity within their natural habitats. Okay, talking about ex situ kind of conservation, where you are doing a conservation of biodiversity, but it's being done outside their natural habitat. And the few examples that you can think of are botanical gardens. It's majorly in fact built up for education purpose, right? Then there are zoological parks. You must have visited zoo definitely at some point of time in your life, right? And then there are seed banks, where all different varieties of the plants are going to be safeguarded. Okay, so now first thing, sacred groves. What are sacred groves? When we talk about sacred groves, you can think of Aravalli Hills of Rajasthan, right? So why are sacred groves examples of in situ conservation? Because they are basically usually a part of forest, which is obviously going to be a natural habitat, right? They are usually a part of forest, and this kind of sacred groves are what are driven by the local people. So as you know that in country like India, where religious beliefs are of utmost importance, due to these religious beliefs and followings that people have over a period of, or people have had over a period of time, these are those particular areas of the forest which, because of religious reasons, are not allowed to be damaged for any reason. And for this reason, we are safeguarding the biodiversity that is existing in those areas. And example I already gave you can think of Aravalli Hills of Rajasthan. Okay, then you go on to biosphere reserves. What are biosphere reserves? They are basically the areas of terrestrial and the coastal ecosystems. Okay, you can think of it this way. And a government also basically tries to safeguard all these biosphere reserves and wildlife sanctuaries, etc., within their natural existing forms by protecting any kind of inhabitation by humans as such, other than the forest department, of course, protection of any kind of wildlife hunting, etc. All that is prohibited in these biosphere reserves and the wildlife sanctuaries. Clear, everyone? So these are few examples, very important examples of your in situ conservation. Let's go on to the ex situ conservation. You have botanical gardens. What are botanical gardens? This is basically those institutes where you're going to list down and document and even have varieties of plants growing within that particular institute or within that particular garden. Okay, you can think of Indian Botanical Garden in Howrah as one of the biggest and the most important examples with respect to India. Okay, then you have zoological parks. Zoological parks, you know what are zoo are the places, basically they are the place, confined area where you are going to keep animals in their restricted area, try to give them a particular area to walk around, to run around, but it's basically artificially created. Okay, then you talk about seed bank. Seed bank is where you are going to have all different varieties of the seeds safeguarded in order to kind of have a bank that just in case of any kind of catastrophe happens or any kind of natural calamity happen, we have the seed bank saving all different kinds of seeds, and then you can use these particular seeds to start cultivating any kind of extinct or endangered kind of plant again. Clear, everyone? So obviously with the given options where we were asked about a method which is not a method of in situ conservation, the right answer is going to be option D, that is botanical garden. Which of the following pairs of gases is mainly responsible for greenhouse effect? Okay, now what is this greenhouse effect? We have been taught about this from our junior classes as a part of our environmental education. Okay, now greenhouse effect is basically a naturally occurring phenomena which leads to heating effect around on the earth's surface and the atmosphere. Okay, now the biggest contributors to greenhouse effect, to this heating phenomena, is basically carbon dioxide and methane. In fact, I'll show you a pie chart which will represent all the percentages of gases which are contributors to the greenhouse effect here. Okay, so the answer here is going to be carbon dioxide and methane. I actually want to ask you to divert your attention slightly to this particular point, which is called as ozone. Okay, many children get confused with the fact that how can ozone be a greenhouse gas when it is actually protecting the earth's atmosphere. Okay, I want you to have a clear idea about what exactly this means. Please think of it this way: ozone gas, depending upon where exactly it is present on earth's surface or in the atmosphere, is going to decide whether it is helpful or harmful for earth. Okay, when this is present closer to the surface of the earth, it serves as a greenhouse gas. It basically contributes to increase earth's temperature. Okay, clear everyone? So ozone is a greenhouse gas, yes, when it is present very close to the earth. But when it is present high up in the stratosphere, it forms the ozone layer, which has a property to absorb harmful UV radiations of the sun. Clear everyone? Would you remember this point? I'll repeat this because this is very important. Once again, ozone, depending upon its location, where it is present closer to earth or far away from earth, is going to decide whether it is helpful or harmful for earth. When ozone is present very close to the earth, it basically serves as a greenhouse gas and has heating effect. Okay, but when it is present high up in the stratosphere, it works like the proper ozone layer that we have been learning about, right, which protects the earth's environment by absorbing harmful UV radiation which are coming from the sun. Clear everyone? So of course this did not have any direct relation with the question. It was not, ozone was not what was asked about in this question, but I still wanted to convey it to you because this is one point where I have seen maximum children get confused. Okay, clear? So obviously the heating gases, like I told you, or the major contributors to the greenhouse effect are carbon dioxide and methane. Okay, so what is greenhouse effect? We are just summarizing everything. It is naturally occurring phenomena that is responsible for heating up of earth's surface and the atmosphere. Clear everyone? Yes. The carbon dioxide gas and methane are mainly responsible for greenhouse effect, and this is the pie chart that I was referring to just a while back. Yeah, you can see this very clearly. Carbon dioxide is responsible almost like 56.4%, and then the next highest is the 16.3%, which is the methane. That contributor to the greenhouse effect is methane. And like I told you, ozone is also a greenhouse gas. Just the point is that you have to understand where exactly is it located, and if at all the question is being asked, question is referring to which ozone. Is it referring to the ozone that is present closer to the earth's surface, or is it referring to the ozone layer which is present high up in the stratosphere? Okay, so the right answer for this question has become option A. Persistent nucellus is in the seed is known as. Okay, so we have persistent nucellus. First thing, what is exactly this nucellus? Nucellus is basically the parenchymatous mass of cell which is present in the ovary. Clear? Yes. So persistent nucellus in the seed is known as. So technically speaking, when the seed development has happened, the nucellus should have been either utilized or kind of have degraded. But there are some seeds which basically have persistent nucellus even after formation of the seed. That is, after fertilization event has happened, after seed has formed, even in that particular seed, the nucellus is going to stay persistent. It's not completely, degraded or consumed. Okay. So here that is called as, what is the question exactly? That persistent nucellus is what is called as the perisperm. Okay, this tegmen, chalaza or chalaza and hilum are quite different terms. They are basically the parts of, tegmen, when you talk about, it's basically a part of the seed coat of dicot seed. Testa tegmen you must have heard of. Talking about chalaza, it's basically the base of the ovule, which of course when you talk about the anatropous ovule is going to be seen, even though it's the base, it's going to be seen at the top. Okay, but it still is the base of the ovule. Then talking about hilum, it is basically the point where the ovule is connected to the main stalk. So that point is what is called as hilum. Clear, everyone? Okay, so let me take you through this whole information regarding this question. Nucellus is that mass of parenchymal cells within the integuments, and it has abundant food reserve. I want you all to remember one point here, that this nucellus is the same nucellus where one of the cells kind of specializes to become the megaspore mother cell, and which will later on give rise to, on meiosis, female gametophyte. Yeah, you remember this point now? So this is the same nucellus. We are referring to this whole mass of cells. Let me change the color of the pen. Yeah, this whole mass of cells that you're seeing here is what is called as the nucellus. Okay, highly nutritive in nature because it has a lot of food reserves as such. And as I told you, one of these cells only specializes to become the megaspore mother cell, which in turn will give rise to the female gametophyte that you are seeing here. Okay, going ahead, perisperm, which is what is the answer to this question. It is basically a nutritive tissue that is found in some seeds that is derived from the nucellus of the ovules. The examples to this are the pepper and the beetroot. Okay, will you remember this example? It's very, very important from exam point of view. So please do remember these examples. Then I was telling you all about testa and tegmen. So these are basically the dicot seed coat, yeah. And the outer one is called as testa. The inner one is called as tegmen. Like you see here, testa and tegmen is what is given in your question. So obviously this is an incorrect answer to the given question. Then going ahead, you had chalaza or chalaza. Like I told you, it's basically the base of the ovule. So if you see this image very clearly, what you're seeing is that there is an ovule like this, which is inverted like this. Yeah, you can see, figure out the resemblance in the structure that is shown in the image and my hand movement. Yes, this basically is the micropyle. This micropyle is where the pollen tube is going to enter carrying the male gamete during the fertilization event. And what you're seeing here is representing the base of the ovule, and that is what is called as chalaza or chalaza. You know, I'm saying or pronouncing both the terms because people use both chalaza as well as chalaza. Okay, clear everyone? Yes. And then coming to hilum, like I told you, the ovule is connected to the stalk by a point which is called as hilum. Okay, this is what is called as hilum. Now here, another point that I want to actually point out is that many children get confused with two terms, that is raphe and hilum. Okay, the difference between the two is that the point at which the main body of the ovule is connected to the stalk is called as hilum. Whereas this fused portion where the ovule body is kind of fused with the stalk or the funicle of the whole ovary, whole ovule, this part is what is called as the raphe. Okay, clear? You have understood the difference. So now please do not get confused with the two. So anyway, coming back to the question, the persistent nucellus in the seed is known as, answer is going to be option C, perisperm. Let's see this question. It's a match the following type of question where you have been given about the ECG. As you can clearly see, those waves that form basically a part of ECG, where you have the recording of the electrical activity that is happening in the heart in column 1, and then the functions that are associated or the changes in the electrical activity that are associated with the particular wave of the ECG is what is listed in column 2. So now we have to create a match. So as you see, we have P wave, we have QRS complex, we have T wave, and then we also have a new thing which is called as inversion of T wave. In what conditions does it happen? Okay, so let us, instead of just trying to create a match here, let us try to recall everything and simultaneously we'll try to attempt this question. Okay, so first thing is, what is ECG? It's also referred to as EKG. It is basically a graphic representation or a graphical record of the electrical activity that is happening in the heart, or the electrical current that is produced in the heart, with which the changes happen in the heart chambers, and this basically brings about the directional flow of the blood in the heart. Clear? Yes. So we'll start with the first one. First one is the P wave. Let me change the color of the marker here once. Yes. Okay. Now here is where you are seeing this particular wave is what is called as P wave. Now this P wave represents the atrial depolarization. What does it represent? It represents the atrial depolarization. It's a very small upward wave, like you can clearly see. And this is when, due to this depolarization, this is when the atria or the chambers, the upper chambers are going to contract. Okay. So this is what is called as atrial contraction, atrial depolarization. Now here, from here onwards, the next complex that comes is the QRS wave. Okay. It's a big wave like you can clearly see. So this is representing the ventricular depolarization, because of which the ventricles contract. Clear? So first what has happened? Atria has contracted when you are seeing the small upward P wave. Followed by that, you have ventricular depolarization and ventricular contraction. Clear? Yes, this is what is representing the spread of impulse in the whole ventricles, right? Then followed by that is a small upward dome-shaped wave like you see here, which is the T wave. This is going to be basically after the ventricles have also contracted, where the blood is going to be pumped to the body. Now the ventricles are going to return back to their normal state or the relaxed state because of the ventricular repolarization. Okay. Yes, this ventricular repolarization is what I'm referring to, and this is going to bring about the ventricular relaxation. Now, sometimes what happens is due to some or the other error or problem, there can be multiple underlying reasons for different heart conditions. What happens sometimes is that there is reduced blood flow to the coronary circulation, and this condition where there is reduced blood flow and hence reduced amount of respiratory gases in that particular area, that condition is what is called as ischemia. Okay, so coronary ischemia basically happens or can be seen by an inversion of the T wave. So like you saw in the previous one, the normal T wave is an upward deflection. But if you see an inversion of the T wave, this basically represents coronary ischemia. Now let us try to create proper match for this particular question. P wave is what? It is representing depolarization of atria. QRS complex is representing what? It is representing depolarization of ventricles. T wave is representing what? Repolarization of ventricles. Inversion of T wave is representing what? Coronary ischemia. So with that, you have understood the right answer for this question is going to be option B. I hope you all got that correct. Following statements describe the characteristics of the enzyme restriction endonuclease. Okay, identify the incorrect statement. So here you have been given 4 statements about restriction endonucleases and their functioning and their action, right? So we have to choose the incorrect statement out of the given ones. The first statement reads, enzyme recognizes a specific palindromic nucleotide sequence in the DNA. So yes, this statement is correct. Why? Because the endonucleases, that is exactly what they do. They have palindromic recognition sites where they make specific cut in both the strands of the DNA. Okay. The enzyme cuts DNA molecule at identical position within the DNA. Yes, this statement is also correct. So this one is correct and this one is correct. Then we go on to statement 3. The enzyme binds DNA at specific sites and cuts only one of the 2 strands. No, like I told you just now, it's going to cut both the DNA, cut at both the DNA strands at identical positions. Clear? Yeah. Then statement D, obviously we've got the right answer, which is option C here. But let us still look at statement D. The enzyme cuts the sugar phosphate backbone at specific site on each strand. Yes, this is a correct statement. What does exactly endonuclease do? It basically cuts or rather makes a break between the sugar phosphate bond in the backbone of the DNA strands. Clear? Now let me help you with recalling all the content that has gone behind this question. When you think of nucleases, what are nucleases? Nucleases are the enzyme which basically cut or break the nucleic acid, right? They basically cut in the backbone of the DNA molecule. Right now DNA I'm using because that is where it is relevant right now. Okay, these nucleases can be of 2 types, exonucleases and endonucleases. Exonucleases, like the name is suggesting, is going to make cuts in the DNA molecule towards the outer end, right? So something like this. Okay, as you see here, you are seeing a double-stranded DNA molecule. where you see that the exonucleases are making cut in both the strands of DNA. This is one important point. Yes, but it is making the cuts towards the outer region of the DNA. We go on to the second type of enzyme, which is called as endonucleases. Specifically speaking, when you talk about restriction endonucleases, their activity is highly specific to only a specific recognition site where it is going to actually cut, right? And these recognition sites usually have a palindrome. What are palindromes? They are the letters, they are the words which are read in same way when you read them from both the direction. Like, for example, this is one basic example that I always give, Malayalam. Yeah. So if you read it from left to right or right to left, it's going to read the same thing, right? The same way when you look at DNA strand, let me give you an example here. Here you are seeing the sequence which is cut by a restriction enzyme which is obtained from E. coli bacteria, and the name of this restriction enzyme is called as EcoR1. E stands for Escherichia, Co for coli, then R is for the restriction enzyme, and 1 is based on the sequence in which it is discovered, right? So EcoR1 is one of the most important restriction enzyme that is actually used in the whole industry of biotechnology. Okay, so this cuts the palindromic sequence GAATTC. So as you see here, we have GAATTC, and in the opposite strand also GAATTC. Yeah, this is 5' to 3'. This is going to be obviously 5' to 3'. So whether you read it from here to here or from here to here, it reads the same. That is exactly what is the meaning of palindrome. And like it was given in one of the statements of the question also, this is going to make cuts between identical sequences on both the strands. That is, it makes a cut between G and A on both the strands. Okay, are you all clear with that particular point? That is exactly what it meant. It means that it is going to cut, this EcoR1 restriction enzyme is going to cut the DNA between G and A of the palindromic sequence on both the strands of the DNA molecule. Yeah, and when this kind of cut is done, usually, now here, this kind of cut is going to lead to production of something called as sticky ends, which basically has, single-stranded overhangs. And the other type of cut that this restriction endonucleases can produce, which is a different endonuclease, of course, those are called as sticky ends. Those are called as blunt ends, right? So like you see here, EcoR1, let me change the color of the marker. Yeah. Like you see here, EcoR1 is a restriction enzyme which has cut between G and A here and G and A here. This has produced sticky ends. So there are these single-stranded overhangs like you see here. Okay, and this basically allows the attachment of, the gene of interest. If I try to put here, it basically allows an easy attachment of that particular gene of interest or target gene. Okay, now the other option that I was telling you about the blunt ends. So enzyme like SMA1, yeah, this is obtained from a bacteria named Serratia marcescens. So SMA1 is basically a restriction endonuclease which produces sticky ends. So which produces blunt ends. What exactly, I'm saying sticky repeatedly. Anyway, so what exactly does this blunt end exactly means? It means that it does not have single-stranded overhangs like the ones that was present in the sticky end or EcoR1 restriction endonuclease generated sequences. Clear? This kind of, blunt kind of ends that are produced by these restriction enzymes like SMA1, like you're seeing here, when we are actually inserting a gene of interest here, we need to also add phosphate groups at the respective ends so that the attachment of DNA to the vector becomes much simpler. Clear, everyone? Yes. So this is exactly the production of blunt ends. It's not given in the question, but this is what is definitely associated with the activity of restriction enzymes, and that is why I have added this point here. Okay, so going back to the question, which of the following statements is an incorrect statement? The answer is going to be option C, because here it spoke about making cut only in one of the two strands, but now you clearly know that it is going to make cut in both the strands. So with that, the right answer for this question is option C. Let's see this question. which of the following is true for golden rice? Okay, so we are looking at golden rice, which is produced in a recombinant way, right? We use recombinant DNA technology to produce golden rice. First thing is, what exactly is golden rice? Golden rice is basically a type of rice which is highly rich in vitamin A content, and hence people who are deficient in vitamin A, or the regions where you see a lot of vitamin A deficiency, if you use this kind of rice there in that particular area to feed the people out there, then obviously the vitamin A deficiency can be easily overcome. Okay, so here let me just change the color of the pen. Yeah. So here what you are seeing, let us look at the options first. We are looking for statement which is true for golden rice. All right, it has yellow grains due to a gene which is introduced from a primitive rice variety. Then there is statement B, which is saying it is vitamin A enriched with a gene from daffodil. Okay, this is a correct statement. So golden rice is actually produced from a gene that is taken from daffodil plant. It's a flowering plant which basically flowers in winters. You know, it's a beautiful yellow colored flower that is produced on daffodil plants. You call them as daffodil flowers also, right? So basically what happens is that this particular plant is highly rich. It has that gene which synthesizes a lot of amount of beta carotene, and this beta carotene is actually a precursor of vitamin A synthesis, right? So that is why the golden rice, when you insert the gene of interest, insert this particular gene which is responsible for beta carotene synthesis, the rice is capable of producing a lot of amount of vitamin A. All right, so statement B is obviously the correct one here. Look at statement C. It is pest resistant with a gene from Bacillus thuringiensis. No, here we are talking about BT cotton actually. If you see carefully, this is what they are referring to, BT cotton, and we are not talking about golden rice here. Okay, so these statements have become incorrect. Look at option D. It is drought tolerant, developed using agrobacterium vector. So let me tell you that the second half of the statement is correct. It is produced using agrobacterium vector, but it is not what is drought resistant. Golden rice is all about having vitamin A enriched content or nutrient. Clear? Yes. So let me take you through the whole summary here. Golden rice is a vitamin A enriched variety of rice which has a gene from daffodil. So like you see this one, this is exactly what is the golden rice. So like I told you, here you are going to introduce a gene which is responsible for synthesis of beta carotene, which is actually a precursor. Let me write it down here. Precursor of vitamin A. And this will lead to synthesis of lot of amount of vitamin A, making it completely vitamin A enriched rice. Golden rice is because of its appearance. You see the color here, it's very, very different from the normal traditional rice. Okay, and the scientific name of, rice is Oryza sativa, just in case you don't know this. Okay, sometimes in the question they can even give the scientific name here. All right, so golden rice, you have normal rice gene, you have a daffodil gene, right? This is what is cloned inside the rice gene, and you have the production of golden rice. Okay, this is the whole total recombinant of the golden rice gene. All right, so the right answer here is going to be your option B. Let's see this question now. So it's a match the following type of question where you have been given about certain type of organisms in column 1, and in column 2 you can clearly see their relationship or how exactly are they existing in nature. So we have to create a match here. We have to talk about saprophyte, we have to talk about parasites, we have to talk about lichens and mycorrhiza. So once again, rather than just creating a match here, let me just help you recall everything here. First thing is saprophytes. What are saprophytes? They are basically the organisms. It can be a plant, it can be a fungus, it can be a microorganism, which are all feeding on decaying or decomposing dead organic matter. Okay, the difference between parasites and saprophytes is that saprophytes live on dead, decaying organic matter. For example, you see saprophytic fungi, and the parasites are the ones which live on the living plant or animals, basically a host. Okay, so parasitic fungi, for example, here you can see an image of a parasitic fungi which is living on the plants. All right, going ahead, you have lichens. It's a very, very, very important ecological association. It's important from exam point of view, yes, but it's very important from the nature's point of view as well. This is basically an association which, allows literally the life to come on land for the first time. Like, if you have heard about this, you have learned about the concept of how lichens are called as literally the pioneers of the terrestrial life that way. Why? Because these are the association which literally allow the life to start happening, to start occurring on the rocks. Yeah, where actually living organisms cannot really survive as such just on the rocks. So what happens is, lichen is basically an association between a photosynthetic component and a heterotrophic component. The photosynthetic component basically makes food, etc., which is represented by the plants. It can be green algae, it can be cyanobacteria, like you see the primitive ones, the smaller ones. Gets associated with fungi, which is the heterotrophic component here. How exactly does it work is that the fungi basically kind of creates a space for itself to kind of grow over hard surface, rocky surface. It can even grow on the bark, etc. But my point is that you can think of the time when the life wasn't there, and when these very primitive structures, these fungi and all, started growing on big rocks, etc. Because of their mycelial growth, they allow the penetration and a good substratum hold to the fungus, and the algal cells grow literally trapped in this mycelial growth. And because of this, because of mycelia kind of forming a network, it absorbs a lot of water and allows the algae to get a support and absorption of water, whereas the algae in return makes food using sunlight by photosynthesis. It's the autotrophic component here, and that provides that food that is made by the plant part of it, by the green algae, by the cyanobacteria part of it, is going to be provided as nutrition to the fungus. So how beautiful of a symbiotic association is this, right? So this is what is called as, this whole association is what is called as lichen. The component which is photosynthetic is also referred to as the phycobiont, because they are usually algal members. So if you know, phyco word refers to the algal algal plants, right? And the fungal component is called as mycobiont. Myco refers to mycelia or fungus, right? Clear, everyone? Then we go on to mycorrhiza. Mycorrhiza is a similar kind of association, but this happens between fungi and the roots of the higher plants. Okay, so this is an association between fungi with the plant roots of higher plants. I want to add this point here. Okay, it's a very specific association. This is also a symbiotic association, where what happens is that these fungus, they grow attached to the root surface of the plant. What happens is that the fungus provides a lot of mycelial growth, of course, because of which a great level of absorption of nutrient and water happens from soil for the plant. So this absorption of water and nutrients is highly essential for the plants to do, right? And in return, what fungus get is that it gets the carbohydrates and the food that the plant is synthesizing. Clear? So this kind of association, mycorrhiza. Rhiza refers to the root of higher plants. Myco refers to the fungal component. So this becomes another kind of symbiotic association, right? Now when you try to create a match, saprophyte, which is saprophyte? Saprophyte is going to be this option. Then decomposition of dead, organic material, it's feeding on that. Then talking about option B, that is parasite, or, column 1. If you see, the second part is given here is parasite. Where do you match the parasite? It's basically living on animals and plants, or any host for that matter. So this is going to be B. Then you look at lichens. Lichens are symbiotic association of algae and fungi. This becomes C. And then comes your mycorrhiza, which is association between plants and fungi, right? So this is going to be your option D. So if you match it, it's going to be A2, B3, C4 and D1, which is basically statement, or rather option A. So the right answer for this question is going to be option A. What would be the heart rate of a person if the cardiac output is 5 liter? So let me mark the keywords here. Heart rate, cardiac output 5 liter, blood volume in the ventricles at the end of diastole, ventricles, diastole 100 ml, and at the end of ventricular systole is 50 ml. Once again, what would be the heart rate of a person if the cardiac output is 5 liter, blood volume in the ventricles at the end of diastole is 100 ml, and at the end of ventricular systole is 50 ml. Let me very briefly first explain you what are these terms. What is heart rate? Heart rate is going to be the number of times your heart beats in a given minute. Simple, yeah? What kind of things affect the heart rate? Obviously, physical activity. This is something that you know, but also the emotional activity. You know, if a person is showing anxiety and palpitations, etc., the heart rate of that person is going to be much faster than a normal person, right? So similarly, these kind of various factors are going to affect the heart rate. Okay, so here we are talking about a person whose cardiac output is 5 liter. What is cardiac output? Cardiac output is the amount of blood that is going to be pumped by the heart in 1 minute. Okay, so that is given as 5 liter. Clear? You are clear with the value that is given here with respect to cardiac output. Now let us look at the second thing that is given. Second and third thing actually. So here we have been given with the blood volume, which is present at the end of the ventricular diastole and ventricular systole. Clear? So with these values, what you need to find out is the heart rate of the person. Okay. So let me take you through certain brief concepts that are involved here. First thing is cardiac output. Like I told you, it is the amount of blood that is pumped by the heart in 1 minute. Okay. So for us, this value is 5 liter right now. Okay. I'm writing it down simultaneously. So cardiac output is going to be equal to heart rate, that is how many times the heart is beating, of course, and stroke volume into stroke volume. What is stroke volume? It is the volume of blood that is ejected from each ventricle due to contraction of the ventricle. So basically in each cardiac cycle, the amount of blood that is pumped is going to be called as stroke volume. Clear? Once again, stroke volume is the amount of blood that is pumped from the ventricles in each cardiac cycle at the end of each cardiac cycle, and cardiac output is the amount of blood that is going to be pumped by the heart after every 1 minute. Like, you know, in total we are talking about 1 minute. Clear? Cardiac output is clear, stroke volume is clear. Now we have to find out the heart rate. Okay, this is easily doable. When you try to check the stroke volume, how will you find out? Whatever is the end diastolic volume. That means when the ventricles have relaxed, they are under diastole, what is the amount of blood that is present there minus the end systolic volume. That is towards the end of the ventricular systole, what is the amount of blood there. Okay, clear? Now we are going to put everything together here. It's a simple application based question where you have to apply the concepts that you know into different numbers that have been provided to you in this particular question. Okay, so cardiac output is 5 liter, which is obviously equivalent to 5000 ml. Yeah. Then you talk about end diastolic volume given as 100 ml, end systolic volume given as 50 ml. Yes. So stroke volume would be what? End diastolic volume minus the end systolic volume. This will be 100 minus 50, which is going to give you the value of 50 ml. Clear? This much is clear. You know the stroke volume now. You know the cardiac output now. All you need to do is find out the heart rate, which I already explained to you in the previous slide that using this formula, we are going to calculate it. Okay. Cardiac output is how much? Heart rate. Yes. The number of times the heart is beating in 1 minute into the stroke volume. What is the volume of blood that is ejected from the ventricles after each cardiac cycle? Clear? So now we are putting everything in the formula here. We know the cardiac output, we know the stroke volume. We just need to calculate the heart rate, and heart rate would be equal to cardiac output upon the stroke volume. Cardiac output is 5000 ml or 5 liter, and stroke volume has come out to be 50, what we have calculated. So the right answer here is going to be 100 per minute. That means 100 beats per minute. Heart rate is going to be 100 per minute or 100 beats per minute, which is your answer D. I hope you have understood how this question is solved, because in exam, if at all you are given different numbers, different volumes, different conditions as such, you would still be able to solve the question using all the information that we have provided you in this particular question. So with that, I'm going to end this question here. The right answer is going to be option D. Let's see this question now. Which of the following statements is incorrect? We are looking at incorrect statement here. The options are: prions consist of abnormally folded proteins, viroids lack a protein coat, Viruses are obligate parasites. Infective constituent in viruses is the protein coat. I think we already know the statement here. Incorrect is what we are looking at. And the last statement which mentions infective constituent in viruses is the protein coat. No, protein coat is merely used for attachment to the host. The infective component is actually the nucleic acid, right? And hence, the right answer for the given question, which is an incorrect statement, is going to be option D. All right, let's quickly revise the concepts involved here. First, we'll talk about viruses. Viruses are basically nucleoprotein, which means what? They basically have nucleic acid core and they are surrounded by specific protein coat, which we refer to as the capsid. The individual building block of capsid is called as capsomere. The genetic material can be either DNA or RNA, and that is what is the infective constituent. And in fact, this is the reason why statement D was incorrect, because it stated that the infective constituent is protein coat and not the nucleic acid. So the right answer, you know, that it's actually the nucleic acid which is the infective one, which is what enters the host cell, which is what, you know, takes over or maybe replicates along with the host cell genome and causes further replication of the virus particles. Clear, everyone? Yes, talking about prions, which was given as one of the options. What are prions? Basically, they are the abnormally folded version of proteins, right? They are basically abnormally folded proteins, and mind you, this is something very, very, you know, these prions can actually cause the misfolding of other proteins also, leading to literally fatal disorders or diseases. For example, you can talk about Creutzfeldt-Jakob disease, right? CJD, you call it as. You have seen talked about, you know, mad cow disease also, which obviously is a very, very, very dangerous kind of neurodegenerative disease. Okay, so that is about prions. Talking about viroids, viroids are much smaller than the viruses. They basically lack a protein coat altogether. They are in fact naked RNA components. All right, very important point. And when you talk about virus as being a parasite, you know that viruses are obligate parasites. They do not really have an existence, a living existence without a host. Only once they enter the host cell, they start becoming, you know, like behaving like living components. So they are obligate intracellular parasites. So the right answer for this question is going to be option D. Let's see this question now. It's a match the following type of question where you have been given certain different parts of the digestive system along with the location where they are present. Okay, or they are the part of the particular digestive organ where it is associated, right? So you have crypts of Libicun, you have Glissen's capsule, you have islets of Langerhans, and then you have Brunner's gland. Okay, this is what you have to do the correct matching with. But of course, like always, like how we solve the match the following type of questions, we'll first try to recall what each one of them is. Okay, first thing, talking about crypts of Libicun. So if you look at the projections, that is villi that are present in the intestine, in between these villi, especially I'm talking about the small intestine, in between these villi are these depressions or invaginations that are there. I'm showing you fingers. Why? Because this helps you understand and imagine what it is like, right? So like in this image, you see these finger-like projections are there, which we refer to as villi, which will bear microvilli in turn, of course. So in between these villi are these folds or invaginations, which are called as crypts of Libicun. Okay, they literally look like simple tubular structures and they are present throughout the small intestine in between the villi, like you see in this image also here. Clear? Yes, this is what is crypts of Libicun. You are going to match it with small intestine. Next is we have Glissen's capsule. Now this name is based on the scientist, right? Now Glissen's capsule is actually a very, very characteristic feature of mammalian liver. Okay, so this is something that can be asked in exam also. Characteristic feature of mammalian liver. All right. The other point about it is that it basically in nature is a very thin layer of connective tissue which covers the whole of each of the hepatic lobule. This is a very important point, right? So this is what is Glissen's capsule. It basically covers each of the hepatic lobule, liver. All right. Next is we had islets of Langerhans. What are islets of Langerhans? They are basically also called as pancreatic islets, yes, but they are basically part of the pancreas, which is the endocrine part of pancreas, what we are referring to. This is responsible for secretion of hormones like, insulin and glucagon, etc., which is responsible for controlling the level of glucose in the body, right? So these are the regions of the pancreas that contain hormone producing cells, like there are delta cells, there are beta cells, alpha cells, etc., right? These are responsible for basically controlling the sugar, the glucose in the body. Yes. Next, we go on to Brunner's gland, which is found in the submucosal layer of the duodenum. And the function of it is the most important point here. The function of it is to produce mucin and alkaline rich fluid, which is very essential to protect the inner environment of the small intestine. Why? Because acidic food from the stomach is going to pass from here. So to neutralize that, to kind of protect the mucosa of the small intestine, internal environment of the small intestine, these Brunner's glands secrete out mucin protein and alkaline rich fluid as well. All right, clear everyone? The Brunner's gland open into crypts of Lieberkühn. So here would be the opening of the Brunner's gland. Okay, so with that, let's try to do the match of it. Now, crypts of Lieberkühn associated with small intestine, Glisson's capsule associated with liver. So after A, I make it B here. Then we have islets of Langerhans, which are obviously associated with pancreas, and Brunner's gland, which are associated with the duodenum. So the right answer is going to be option D here. Let's have a look at this question, a very important question, okay? Slightly detailed in the concept here. Which of the following immune responses is responsible for rejection of kidney graft? Okay, so actually in this question, what is being asked about is a kidney graft, but you have to understand that we are talking about any graft in general. What is a graft? Something that has been taken from some other part of the body or maybe some other person altogether and grafted or transplanted in an individual. Okay, organ transplantation is one of the best examples for this topic to be understood with. Okay, so here the options are cell-mediated immune response, autoimmune response, humoral immune response, or inflammatory immune response. Okay, so the right answer actually here is cell-mediated immune response. I'll speak about the incorrect options as well, but first let me actually take you to the cell-mediated immune response. What is exactly the cell-mediated immune response? This is a type of immune response which is mediated or carried out by specific type of cells of the immune system, which are called as T cells. Okay, T cells. The immune response, which is mediated with respect to fluid mechanism, or with the help of circulating antibodies, etc. That one is called as humoral immunity. Okay, clear everyone? So active infection, etc., that is majorly taken care by the humoral immunity, where obviously both these immune responses are going to be working in coordinated fashion with each other. But yes, when it is about the circulating antibodies, etc., then it is called as the humoral antibody, which is mediated by the B cells. And here what you are seeing, the cell-mediated immune response or cell-mediated immunity, this is being done by the T cells. Okay, so here T cells are majorly of two types here. There are actually more types of T cells, but right now we are only talking about the relevant information here. We are talking about the cytotoxic T cells, killer T cells that is, and the helper T cells. As the name is already suggesting to you, helper T cells are the ones which will help mounting of other type of immune response by activating other cells, and this is usually done with the help of special cell signaling molecules which are called as cytokines. Okay, what are cytokines? They are simple cell signaling molecules. One cell gives the signal to the other type of cell that, listen, you need to get secreted and start doing your work. Listen, you need to get secreted now and start doing your work. So this kind of thing is what is done by the cytokines. Okay, clear everyone? Talking about the killer cells or the cytotoxic T cells, these are the ones which are going to kill the infected cells. Whether they are self cells or any type of cells, these cytotoxic T cells are the ones which are going to bring about literally the killing of the infected cell. Clear everyone? Okay, now we'll talk about the whole pathway of how exactly this works. First thing, the T cells respond to the antigens which are located on pathogen. Okay. Now here I am talking about a general mechanism and not just a mechanism where you are talking about organ rejection, etc. We are talking about in general the mechanism of the T cell functioning. So what do T cells do? They respond to the antigen that is located on pathogens. What are antigen? They are specific surface receptors that are going to be present on each different type of cell. We have specific receptors present on each different type of cells of our body. Similarly, pathogens also have the same. Yeah, they have specific proteinic molecules on their surface. Okay, so T cells respond to this first. The cells called macrophages, these are basically specialized phagocytic cell. Macro means big. Phagi refers to cell eating, right? So these are basically cells which are specialized in phagocytosis. So cells called macrophages displays the antigen on an area called MHC protein, which refers to major histocompatibility complex. Okay, let me just tell you in brief about MHC. MHC is basically the set of receptors that are present on the cell, which allows the cells, the immune system cells of our body, to understand which one is a cell that is belonging to the self and which one is a non-self. So this differentiation between self and non-self is done with respect to MHC, with respect to major histocompatibility complex, which is basically nothing but a set of receptors present on the surface of the cells. Okay, whenever MHC comes to you, please understand it is relevant to self and non-self differentiation, of course, and it also helps in, of course, antigen presentation. Okay. Now, the T cells connect to the antigen because the antigen has been presented literally to the immune system cells. The macrophage respond to the connection by producing cytokines. Now we have started producing the cell signaling molecules. Yes, what did I tell you about cytokines? They are the signaling molecules. So these cytokines will now bind to the surface of T lymphocytes, which will activate the T cells to produce its copies, its clones, and further bringing about this whole cascade. Okay, further bringing about this whole cascade of events where it is going to ultimately lead to the lysis of either the pathogen infected cell or maybe the foreign organ cell which is going to be rejected, etc. Yes, clear everyone? This is like a beautiful summarized version of what I just spoke about. Example is, obviously we have taken the example of transplanted organ like kidney graft. It can be a liver graft for that matter, where the body is going to function in a very similar way with respect to the immune response. Clear everyone? Yes. Okay, now we had incorrect options also. We had 3 incorrect options to the question. One was given as autoimmune response. Example of autoimmune response to is going to be rheumatoid arthritis, where body's own cells are attacked by the immune response or immune system for that matter. Okay. The other thing that was given was humoral immune response, which I kind of already explained to you. It's controlled by the activated B cells and the circulating antibodies. For example, viral or bacterial infection, pathogen-infected cells can stimulate B lymphocytes to produce antibody against the antigens. And these antibodies are going to be produced by specific plasma cells, which are, circulating in the body. And they also lead to differentiation into the memory cells, because of which a secondary infection with that particular virus or that particular bacteria or that particular pathogen, the secondary response becomes very, very quick. It means what? When the second time the infection is happening with the same pathogen, the body is going to react much faster in comparison to the primary response, the first time the body had responded. Clear everyone? And then there was another incorrect option which said inflammatory immune responses. So this is not what is directly involved in the organ rejection. Here what happens is that the body produces inflammation, including redness, heat, swelling, and pain, etc., which happens as kind of a generic mechanism in order to prevent any kind of infection that is happening, or even allergy for that matter. Okay, for example, like you see the swelling that happens after mosquito bite, or in fact any insect bite. If you see, sometimes what happens is that the skin swells up, it becomes warm. If you try to feel the temperature next time you get an insect bite, try to see the temperature of that particular swollen red area. You will feel it is much warmer than the other areas. Why does this happen? This happens especially because of histamine, which causes the blood flow to really increase, to migrate all the cells to that particular site of biting, etc., so that the body produces inflammatory responses against the pathogen, or maybe against the insect bite, or whatever changes are happening to the immune system. Okay, so coming back to the question where we were asked about rejection of kidney graft or any graft for that matter, the immune response that is involved here is going to be cell mediated immune response. That is option A. Identify the cells whose secretion protects the lining of gastrointestinal tract from various enzymes. So we are looking at the specific type of cells out of duodenal cells, chief cells, goblet cells, oxyntic cells. Which one of these are going to secrete out something which protects the lining of GI tract? The answer is straightforward here. You know what are the unicellular glands which secrete out mucus? It's the goblet cells, right? It's the goblet cells. Talking about all the remaining options also, we'll come to goblet cells as well as, duodenals and chief and oxyntic cells, etc. First is goblet cells. These are also called as the mucus cells. These are present throughout the epithelium where they secrete the mucus which protects the lining of gastrointestinal tract. This is very important because, as you know, all across the digestive system, there is a lot of change of the pH that is happening, right? Stomach is highly acidic in nature, and to neutralize that, then the small intestine secretes out a lot of, alkaline juices, etc. So all these various drastic changes of pH are happening, and hence it's very important for the whole lining of the gastrointestinal tract to be protected and to be kind of, taken care of. And this is what is done by mucus that is secreted by the goblet cells. Okay, now you talk about duodenum. Brunner's gland are there, which basically produces something called as mucin. This is very important. The mucus protects the duodenal wall from getting digested. This is a very important point because it basically kind of contributes towards the alkalinity found in the small intestine. Okay. Then we go on to the chief cells or the peptic cells. These are basically present in the gastric gland. What are they going to do? They are basically going to synthesize the inactive precursor of an enzyme, which is the zymogenic form. What is zymogen? It's the inactive precursor of an enzyme. And the example here you can think of is pepsinogen. As you know that the peptic cells or the chief cells are responsible for synthesizing pepsinogen, which will obviously be activated later. What is pepsinogen? It's the protein digesting enzyme, but in the inactive form. Pepsinogen on activation will get converted to pepsin. Okay. So then you would remember the whole of the events that is happening in the stomach and the intestine, etc. Okay. Then we go on to the oxyntic cells, also called as the parietal cells. These are responsible for synthesis of HCl, hydrochloric acid, and Cassell's intrinsic gastric factor. What is HCl doing? First thing, HCl is activating the pepsinogen that we just spoke about. It is also going to kill all the microbes that are existing in the food that has reached the stomach. Other important point about the Cassell's intrinsic factor is that this basically helps in activation, or rather I should say absorption of vitamin B12, which is a very important point. Clear everyone? So the right answer for this particular question is going to be option C, goblet cells. Under which of the following conditions there will be no change in the reading frame of following mRNA molecule? Okay, so we have a sequence here, 5' to 3' sequence of mRNA, very important point. And we are looking at the condition which will lead to no change in the reading frame. Okay, first thing is, what is reading frame? When you try to carry out the process of translation or protein synthesis, we all know that it's going to start with something called as a start codon, which basically is located at the 5' end of the mRNA, the functional mRNA we are talking about. So when the translation is going to happen, it is going to happen from that particular start codon, and from there every 3 bases is going to be from there on till we reach the stop codon. Every 3 bases, which is going to be referred to as codon, is going to be responsible for insertion of that particular amino acid, which is coded by that codon. All right. So we are looking at that reading frame. We are looking at a condition where this frame is not going to change. Start to the stop codon frame is not going to change. Okay. So let us look at the options here. Statement A is deletion of GGU from 7th, 8th, 9th position. So here we are bringing about changes in 3 bases. This is an important point and an important hint that you can think of. Option B, insertion of G base at 5th position. G, basically we are referring to a single nucleotide here. Okay. Option C is deletion of G from 5th position. We are looking at another point where we are bringing about a change in a single base pair. And then option D, insertion of A and G at 4th and 5th position respectively. We are looking at A and G. That means we are looking at two bases being inserted at a given position. Even if it was deleted, it would have, been a similar kind of change. I'll tell you why. Because if you think of it, you know that when you talk about the reading frame of an mRNA molecule, you would be reading sensibly by reading 3 bases. Okay, how do you read an mRNA, or how does the enzyme read an mRNA? It reads it in a sequence where it is going to read the first 3 bases, then the next 3, then the next 3, and then the next 3. So which out of the given condition is going to make the minimum change here? Only when you are going to remove out either 3 bases or maybe insert any 3 bases, because of which, as it's in the multiple of 3, or rather 3 or multiple of 3, it's going to bring about the minimum amount of change in that particular sequence. If you insert a single base, the whole frame is going to change. Not only just that codon, it's going to change the whole of the codons downstream. Also, if you delete a base also, whether it is a single base or maybe 2 bases are inserted or deleted, it's going to change the whole reading frame downstream from there. Whereas when things are happening in multiples of 3, because as you know, a codon is a sequence of 3 bases. So either you insert 3 or delete 3, that is something that is going to make the minimum difference. Okay. Clear, everyone? Yes, now let's try to apply everything that we have learned in this particular question. So, gene mutation, I'm starting with gene mutation. What is gene mutation? It is a change in the expression of gene due to change in either number, sequence, or the type of nucleotides. Based on this, it can be any different type of mutation. When we talk about frame shift mutation, it is one of the varieties of gene mutation. What happens is that the reading of codons is changed due to either insertion or deletion of the nucleotides. Okay. Okay, so here we are talking about this particular sequence. This is the same sequence that we have been provided within the question, and this is where I have written it down. So 5' AAC, AGC, GGU, GCU, AUU, 3'. This is how the mRNA of the given question is going to read. The first, one of the option, not the first option, but one of the options was insertion of G at 5th position. Let us try doing this. We have inserted this G base at the 5th position of this particular mRNA. What will happen? You see that codon 2 has changed completely. Yes. Codon 3 was GGU. Now it has changed to CGG. Codon 4 was GCU. It has changed to UGC. Next one, AUU, has now changed to UAU. Like I was telling you, a change in single base addition or deletion is going to bring about a drastic change. All the things downstream are going to change here. Yes. So this is not a condition where, the minimum change is seen or no change is seen. Okay. Then the next option was deletion of G at the 5th base. So it's the same story here. We are deleting the G from the 5th base. So what will happen? Everything would shift towards the 5' end. So AAC, the first codon, is going to remain the same here. The second one, because G has been removed from here, it has become ACG now, which was AGC earlier. GUG now, CUA now, and then UU, and further down the sequence, whatever would be there. So you've understood, this has brought about a change in all the downstream sequences again. Okay, so this option also will bring about a drastic change in the given mRNA. Let's talk about insertion of A and G, that is 2 nucleotides at 4th and 5th position respectively. So we are inserting, this is the 3rd position and this is the 4th position. So obviously between them is the 4th and 5th position. This is where insertion is going to happen, right? So after AAC, we have inserted A and G. What is the change that we are seeing here? So first codon is same. The second codon has become AGA. The third codon has changed. 4th has changed. 5th has changed, and so on. Right? So what you are seeing here is that due to insertion or deletion of a single or 2 bases, we are seeing drastic changes that are happening here. Let us try and delete 3 sequences, rather 3 bases from 7th, 8th and 9th position, like GGU, how it has been mentioned in the question. So what we are doing is we are removing out bases in the three number, or in multiple of 3 also if we do, we are going to see the same change. So as you see here, codon 1 has remained same, codon 2 has remained same, codon 3 has been deleted altogether. So codon 4, that is GCU, you see it here. Codon 5 AUU, you see it here. The only change that has happened is in a single codon. Okay, so if the number of nucleotides inserted or deleted is a multiple of 3, then there will be no change in the reading frame as such. Every time you would read the similar way downstream in the given mRNA in a similar way. The codons would be read in a similar way. Clear, everyone? So the right answer for this question is going to be option A. This is a very, 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 very important and a question where you can definitely sure shot get full marks only simply by knowing the concept here. Okay, so the right answer for this question is going to stay as option A. So children, that was all about the discussion of today's set of questions. I hope you all have followed all the concepts that were included in today's question set, right? Go back, try and practice to understand what is, the concept that is involved in that particular question, so that you never attempt a question wrong based on that particular concept. We'll meet up again with the next set of questions. Till then, keep learning.